Thank you so much for the introduction. I would like to start with you having to imagine a world where we would be unable to use emotions in our everyday communication. Let me give you an example. If I was talking like this for the next 10 minutes, you will have a very hard time following me. The reason why you might feel like this is because I'm not using emotional expressions in my speech to make my words effective, or in other words, salient. My name is Thomas Pomberger. I'm a neurobiologist in the lab of Dr. Richard Mooney at Duke University, and I study a cortical circuit for communicative salience. This circuit is located in a brain region called the insular cortex, or also called just the insula. What you see here is a schematic depiction of the location of the insula in the human brain. The insula plays a role in many different behaviors and functions. Three of them are emotion processing, multisensor integration, and salience detection. In my research, I'm interested what the insula's role in social vocal communication is. To study this, I'm using mice as an animal model. Mice tend to produce so-called ultrasonic vocalizations when they communicate with other mice. We humans cannot hear those ultrasonic vocalizations, but we can look at them through tools like, for example, here, a spectrogram. You see here an example bout of a mouse of ultrasonic vocalizations. To record insular activity during these vocal behaviors, I'm using a method called miniscope calcium imaging. Miniscopes use genetically encoded calcium indicators to measure calcium activity when neurons are active in the brain. Now, with this method, the most robust way to elicit ultrasonic vocalizations in a mouse is when a male is vocalizing towards a female during courtship behavior. What you see here is the beginning of a video that shows a mouse, a female mouse, with a male mouse that is carrying a miniscope. And I will start this video in a few seconds, and you will see the male courting the female. But also listen carefully. So you can see the male is investigating and also chasing the female, and you can also hear his vocalizations. I downshifted those into the human hearing range, and usually you would not be able to hear them. So with this very robust behavior in mice, I designed the following experiment to study different social vocal contexts and what the insula's role is in them. So what I did is I developed a two-chamber assay, and I placed a mouse, a male mouse, with a miniscope in one of the chambers. In the first condition, this mouse was paired with a female, and in the other chamber, I placed another male. Now, the mouse with the miniscope, the male, started to vocalize towards the female while I recorded in the insular cortex. In the second condition, I removed the female from this male and paired it with the other male. So now this male started to vocalize towards this female, and the other male, which we are recording a neural signals from, is now actively eavesdropping on those other males' vocalizations. And then finally, I removed the female and the male and used only playback of ultrasonic vocalizations while recording in the insular cortex. Here's what I found. So what you see here is neural activity in the insular cortex in a male that is vocalizing towards a female. So what you can see here in the middle panel is many hundreds of neurons and their average activity aligned to ultrasonic vocal bout onset, depicted by this dashed line. You can see in yellow that there is many excited neurons, and in blue, there's also a bunch of suppressed neurons during this behavior. Below, you see those two populations, the excited ones and the suppressed ones. And their overall population activity. Interestingly, you can see that the activity of both populations starts to ramp up before vocal onset. And we believe that this indicates that there's a premotor signal encoded in the insular cortex. But what about the other conditions? So when I remove the female and pair it with the other male in the eavesdropping condition, we see that an almost non-overlapping different subset of neurons becomes active right after vocal onset of the other male. So there are two populations that are largely distinct, one encoding vocalization and one encoding listening to another male's live vocalizations. Now, however, when we only use playback of ultrasonic vocalizations, we barely see any activity. But why is it that we see this difference in eavesdropping versus playback? And to answer this question, my colleague Kate Kaplan and I performed the following experiments. So we had fixed mice and trained them to run on a treadmill while we recorded from the insular cortex and used ultrasonic vocalization playback. We then did the same experiment again, but this time we used airflow to deliver female odor to the same male while playing the same vocalizations. And here's what Kate and I found. So you see here the overall average population activity of the population of excited neurons and the suppressed one. On the right are the suppressed one, on the left are the excited ones. And those neurons you can see in two different colors on each plot. One curve is without female odor, and the other one is playback with female odor. And what you can see in both cases that the overall amplitude of this population activity is significantly higher when female odor is delivered during playback. This shows that female odor plays an important role in how the insular cortex encodes socially salient vocalizations. We also were interested in how the insular cortex is connected to the auditory system as well as to the vocal motor system. And to test this, we used some viral tracing methods. So what I did here was I injected a virus into the auditory thalamus, shown here as MGB. And this virus expresses ChRE recombinase in infected cells, but also jumps postsynaptically, which means that if the insular cortex receives direct projections from the auditory thalamus, we should also have ChRE recombinase expression in these insular neurons. At the same time, I injected a color flipper in a vocal gating region called the periaqueductal gray in the midbrain. This color flipper virus infects neurons at the axon terminal and travels retrogradely and then expresses a red fluorophore in infected cells. However, if those infected cells also express ChRE recombinase, the fluorophore shifts from red into a green fluorophore. And indeed, when we do histology after a few weeks, we see green cells in the insular cortex. This shows that the insular cortex seems to directly link the auditory thalamus with this vocal gating region in the midbrain. So what I showed you in the last few minutes was, first, we were able to show that there are two distinct populations in the insular cortex, one encoding active vocal production and the other one being active when the same male is eavesdropping on another male's vocalizations. Then we were also able to show that female odor seems to be very important in encoding those live vocalizations. And finally, we were able to show that the insula directly links the auditory system with the vocal motor system. Now, for my future research, I would like to dig a little bit deeper into the different cell types that encode this different context of social vocalizations. Equally important, I'm also very interested in what role neuropeptides such as oxytocin play in this different context and how they shape these social vocal communication behaviors. With that, I want to thank the organizers of the Next Generation Faculty Symposium. I'm really grateful having the opportunity to talk here. And I also want to thank my former and current colleagues, supervisors, and mentors. And I hope I used enough emotional expressions in the last minutes so that you didn't lose interest and that my talk was salient enough for you. Thank you.